So today, when we talk about disruption, we talk about computers and IT. Because this morning, when I woke up, like my phone woke me up, and I didn't even have to go out of bed and I could check my mails. And sometimes I catch myself texting friends, even if they are in the same room than me. And this tells how much IT is disrupting our lives every day. And still today, as Pascal said, we have disruptive technologies that change the most conservative industries, such as taxis and hotels. So all started with an idea, the idea that a code could be executed by a machine, and that this machine could perform tasks for us. But what we really forget so often is that it took an incredible amount of effort to reach to where we are today. We started with computers that were reading perforated cards, to the extent that I cannot even say it properly, to develop electronic computers that were filling entire rooms. And then came the real disruption, the personal computers. It was the first time that computing could be brought to society. And then the rest of the story, I'm not going to explain you, because not so long ago, we could take our computers anywhere and work from everywhere. And now we have mini computers in our pockets that do any kind of task, and we can even not live without it anymore. But what I would like to talk about today is a revolution in a field that we hear less about. It is biology and medicine. And the trigger of this revolution was, exactly as Pascal said, the fact that now we can decode human genomes. And more than decoding human genomes, we can actually edit it. But we are running into a conceptual problem here. We can decode the code of life, we can edit it to do whatever we want, but we have really difficulties to understand what changes we do in the genome, what turns out for patients. And right now, we are using animal models. These animals are great because we can take their genomes and understand their biology. But as our society is evolving, we are aging and we are facing more complex diseases, such as cancers, metabolic diseases, and neurological diseases. And these models are not sufficient anymore. They cannot represent our biology. And the example that I have in mind is a few months ago, a company developed a new painkiller. And everything was going fine until they administered it to patients. And some of these patients started having severe brain damages. And we didn't see anything in animals. We couldn't predict that. And this shows you how much these models are insufficient to address our current diseases. So you're going to tell me, okay, but how can we represent humans outside of humans, right? It's not that trivial. And the answer is surprisingly very singular. Since the 1950s, we've been trying to take human cells and make cell lines outside of our bodies. And we were culturing these cells in a technology that was invented in 1887, the Petri dish. But what is more impressive is that still today, we are using the exact same technology to culture our cells. So I'm going to ask you to take a step back here. So basically, when I graduated from an hybrid college, I studied both engineering and biology. And when I started working with these models, I thought to myself, but this is not possible. I mean, we cannot represent humans with just plastic surfaces, right? I mean, that doesn't make sense. It's a bit like you would ask computer engineer now to develop the most complex code you want and run it in the first electronic computer. So I want to tell you something. I want to tell you about our biology. So basically, all of us are a collection of organs, and each of these organs are a collection of cells that do specific jobs, right? And what is so interesting about our biology, and also animals ones, but especially our biology, is that within each of these organs, there is a specific type of cell which can actually replace our dying cells, and we call them stem cells. But what is lesser known is that when we try to take these cells and put them in our 130 years old technology, we lose them. They don't function like they function in our organs. And I think you can appreciate with me that there is a fundamental difference between these 2D plastics and our bodies. One of the main ones is that these cells are cultured in 2D, whereas our organs are 3-dimensional. And these cells, these stem cells, are surrounded with many other cells that can keep their function in our bodies, whereas it's not the case in these very simple platforms. So biologists then started thinking, okay, we need to solve that. And it's by asking other fields such as material science that we started being able to culture our stem cells in 3D. So we started developing gels that we called in the jargon hydrogels, but you can imagine that a bit like gelatin in your kitchen, that have all the necessary components for growing and making these stem cells survive outside of our body. But what is the most interesting in this environment, these gels, is that these stem cells don't only survive, but they also produce all their neighboring cells they need to create an organ function. So it means that now we can have stem cells that keep stem cells and that actually represent functions of organs outside of our body. And let me walk through a few examples because it's really impressive. So one of the most striking one is the cerebral organoid, and that the media like calling mini brains, right? So what is really striking with these models is that when you take stem cells and you grow them in these 3-dimensional cultures, they start growing, producing their neighboring cells, and organizing exactly like how they are organized in our brains. And this is not the only example. Because besides these models that we can use, these mini brains we can use for modeling complex diseases such as Alzheimer's, schizophrenia, there are other models that have been developed that create cells from the retina. And with these models, we can develop drugs against very complex diseases such as macular degeneration. And ultimately, we are going to be able to cure blind people with these cells. Also, we can create mini pancreases in vitro that produce insulin-like cells, insulin-producing cells, to address the problem of diabetes. And finally, we can take intestinal stem cells and grow these mini intestines to address cancers and also inflammatory bowel diseases. So this is the first time in biology that we can take our stem cells and grow mini functional organs outside of our body. But I want to tell you why it's even more exciting than what it is right now. It's because the following. So less than 10 years ago, there was a technology described by a Japanese professor that's called induced pluripotent stem cells. And what it means is that you can take any one of us cells, like a blood cell or a skin cell, and you can retransform them into a stem cell. And these transformed cells, these stem cells, can look a bit like embryonic cells. They can do whatever cell of our body. But what is really amazing is that from these cells, we can generate mini organs of every one of us. And this gives us new amazing opportunities because now we can understand these complex diseases. We can understand why in one patient this drug works and in the other patient it doesn't work. We can now develop new treatments for like neurological diseases and all these things that we don't understand at the moment. And in the end, we are going to be able to cure uncurable diseases with stem cell transplantation. So hand in hand with genetic profiling, this technology is going to be able to make personalized medicine, through personalized medicine, this dream come true. But what I haven't told you is that this technology is very young. And at the moment, it is possible to make these structures in very limited labs around the world. It's a bit like we are at the same stage that the personal computer was when it was discovered. We need a really tremendous work to make this useful for society. And it's going to start with the engineering of new materials, clinically relevant, to put these structures ready for the clinics, to merge this field with microfabrication, microtechnologies, to miniaturize this technology and put it in the pharma. We are going to need the help of chemistry and physics to understand these structures, to see if really they represent our body and also to which extent. And in the end, we are going to need computer sciences and information technology to analyze this data, store this data, and make something comprehensive out of it. And I want to leave you with this, because this problem is not just putting several fields beside each other. It is actually creating hybrids, people that have a new way to look at problems and that will invent the future that we cannot even imagine tomorrow. And so if you're a university head or a company management head, push your young generation to take their expertise and merge it with other fields that they are not used to. And that's how you're going to be able to promote innovation. And if you're a student or finalizing your education, you can really explore other areas, something totally unrelated, and you can merge that with your field and be able to be the innovator of tomorrow. And with this, we are going to be able to all surf together the new wave of the new revolution, which is going to be the revolution of medicine and healthcare. Thank you.